I'm Brandon Staglin. This is One Mind Brainwaves, where we talk with leaders who are making waves for brain science and mental health. I'm excited about today's show. We're welcoming one of One Mind's newest Rising Star awardees, Dr. Xin Jin of Scripps Research Institute. She'll be talking about her great advances recently in her work in genetic screening for mental illnesses. I also want to give a great shout out and thank you to Compass Pathways for their amazing sponsorship and generous support of all this year's Rising Star awardees, including Dr. Jin. Thank you, Compass Pathways. But first, One Mind's annual music festival for brain health was held earlier this month, at which Dr. Jin spoke, amazingly. As in past years, hundreds of passionate brain health advocates came together for a fabulous day of food and wine and music, and science, of course, was the star. The music festival celebrates and raises funds to support the work of groundbreaking scientists like Dr. Xin Jin and the innovative research that they do. Today's guest Dr. Jin is an assistant professor of neuroscience at Scripps Research, where she's also a member of the Dorris Neuroscience Center. She recently was named a 2022 One Mind Compass Pathways Rising Star awardee for her groundbreaking work in using genetic screening to understand and predict the risk for mental illnesses. Xin, thank you so much for being with us today on Brainwaves. Welcome.
Thank you. They're happy to be here.
Awesome. Great to see you. And viewers, don't forget, we want to hear from you too. If you have any questions or comments you'd like to post in the chat today, please go ahead and do that. And if you know of anyone who might benefit from the information we're sharing today, please share this webcast with them. Thank you. Okay, well, Xin, let's get started. I'd love to learn more about your work. You know, in recent years, a lot of progress has been made in identifying genetic risk factors for mental illnesses, psychiatric disorders. And what can we learn from genetic screening, actually? What are the ways that genetic screening can help us learn about how to predict or treat, even better treat mental illnesses?
Yeah, I think that's a really fantastic question, and also very exciting direction that many, including our labs, interested in working on. In the past, I would say almost 2 decades, now we know many of our sort of risk in manifesting psychiatric disorders actually strongly associated with the genetic factors. We started with about 1 or 2 genes about 10 years ago, but currently we have almost 250 genes with strong significance associated with mental illnesses such as autism, schizophrenia, and so on. So now I think it's an exciting time to think about understanding these genes and the actions they make in the cell can offer us an opportunity to understand the mechanism of psychiatric disorder, which is one of the most complex and devastating disorders. But at the same time, I think an obvious question is that there are so many genes out there, and they are very different, they are very diverse, they are different genetic risk variants, but obviously at some point they converge into this pathways or mechanism that gave us, you know, one of these very few sort of so-called phenotypic manifestation of the disorder. And the question is why and how? How can we study hundreds of risk genes quickly, systematically, and also unbiasedly to understand what are they really doing? And hopefully when we're looking at 200 risk genes, they are not doing 200 different things, but they're obviously not doing one thing. So how do we actually come to this platform to resolve at high resolution to read out what their cellular actions, but also figure out a way to do this scalably, not looking at 1 or 2, but hopefully dozens or even hundreds of them at a time. And I would say the limitation that we have in the field collectively at this point is probably this sort of like dilemma between resolution and scale. Because we have been studying one gene at a time for many, many years or even, you know, decades. But with the question of neuropsychiatric disorder and the diverse genetic variants, studying one gene at a time is no longer enough. We want to do it scalably. And for many of the risk genes, we don't even know the very fundamental, such as among all the brain regions, what is the brain region that's manifested in the disorder, so-called autism spectrum disorder, among all the cell types, including neurons and non-neuronal cells, which are the cell type that's more vulnerable, that's impacted by these disorders. And also within the cell, what are the molecular pathways and network that's impacted? These are very fundamental questions that we actually don't know. And we definitely do not know for a long list of genes. And collectively, hopefully in 20 years from now, we'll be able to make some progress.
Well, it's tremendous. It sounds like the work that you're focused on aims to not only understand, is not focused on identifying new genes, but like understanding what the genes do in the brain that lead then to mental health challenges in people, right?
That's right.
Yeah. Okay. That's at scale, doing lots of genes at once, and then at a high resolution. It sounds like, so you can get a lot of precise information about what the genes are doing, which could lead to better diagnostics, better treatments for people who are struggling, right?
Yeah, absolutely. What we're hoping to do is this balance between the resolution and scale, right? How do we have the cake and eat it too? How can we get the scalability, but also reach high resolution readout? Absolutely.
So it sounds like you have some innovative ideas about that. Can you explain how your research will advance this field and take the science in new directions?
Yeah, totally. So a few years ago, we started, precisely thinking about this question, about how to establish a high resolution readout to study gene function, but also with scalability. How can we have the best of both worlds, per se? So we started building on this platform called PerturbSeq, that is basically combining the in vivo CRISPR-Cas9 gene editing technologies, which is a way to precisely introduce changes in the genome. But we're also using a high resolution readout called single cell RNA sequencing, which is we can dissociate the cells and isolate cells and analyze the cells one by one. So for each of the cells, they might receive one of these very different genetic variants that's previously we discussed that implicated in neuropsychiatric disorders. But instead of taking one sort of unit as an animal, now we can actually understanding these genetic mechanism one cell at a time. We can actually introduce these, we call it pool perturbation, because we mix all the different CRISPR cocktails, so that within one brain, there are actually many different clones and cells that receive different perturbations. And afterwards, we can analyze these perturbations in each different distinct cell types one at a time that allow us to ask, with this perturbation of this gene number one, what are the cellular changes in this particular cell types? And we can do these with scale, meaning we can harvest tens of thousands or hundreds of thousands of cells very easily and analyze much in depth what are the cellular changes results in these genetic changes and genetic perturbations. So this is the technology that we published a few years ago called In Vivo PerturbSeq, which we first applied to look at a few of these risk genes implicated in autism spectrum disorder. And of course, we're very fortunate to have the support from the One Mind Foundation, which we will strengthen our platform and look at additional brain regions, additional cell types, and also in additional developmental windows, just to see what are really the cellular action from space to time that they can manifest in distinct cell types.
This is incredible. It's like a one-stop shop to understand what the genes are doing in the cells and the molecules at a very precise level at a large scale. What a great breakthrough this will be, and is already with the work you've done so far. For our listeners, can you break down a little bit some of the terminology, like what does in vivo mean, please?
Yeah, so we're interested in doing this experiment in vivo, meaning these are in a living animal. One of the reasons that we really care about in vivo studies, especially related to psychiatric disorders, is that we know that our brain is a very complex network. It's not just, 100 millions of very smart neurons on the Petri dish on two-dimensional space, right? We are intelligent because our neurons have this very intelligent way of connecting with each other. They are influencing each other. And when it comes to diseases and disorder, I think it's also very important to think about the disease and disorder usually is not coming from just one cell, but rather the network their organization, their connectivity. So we try to do these experiments from intact tissues. In this particular case, both in vivo perturb-seq and perturb-map are basically experiments done with intact tissue without having to sort of worry about the differences of cells culturing on a petri dish versus the intact brain that you and I have that hopefully generates thoughts, behaviors, and so on.
So it's a more kind of like realistic environment to do this testing in and understand better what's really going on. You can then translate more directly into understanding what's happening in human brains, it sounds like.
That's definitely our hope.
Yeah, excellent. So, during your relatively short career, you've already established yourself as a leader in this field, a true expert. What first drew you to this area of study in neuroscience and in particular genetic screening?
Yeah, that's an interesting question, a funny one, because I actually didn't quite start my career in neuroscience. I started my training as a chemist, actually. And I think I'm not alone because many people in the neuroscience community, and I think it's a wonderful thing, they all come from very different paths. Some of them were trained like me from chemistry, from physics, but I think that's really the strength of the community because we can all come from different angles and perspectives, bringing very different expertise and to study this rather complex organ, the brain. And for me, I started building chemical biology tools in a chemistry lab. And later on, I think little by little, I started wondering what are these tools that I'm building can be helpful to study. And that's when I started working on cell biology and I started my PhD. That's my first time sort of working in neuroscience in the lab of Corey Bargman, studying C. elegans, which you might think it's a mini brain with only 302 neurons compared to the brains that I'm studying today. It was rather compact, but at the same time, it's very logical and defined because even 10 years ago when I was almost 10 years ago when I was studying my PhD in C. elegans, such a simple model organism, there are very strong genetic tools and they have a very well mapped nervous system. We know precisely the 302 neurons. We even give them names. So I think training in this sort of chemistry as well as the compact nervous system really gave me this drive that I want to bring the clarity and the precision from these simpler nervous system or a simpler synthetic system into the obviously more complex mammalian nervous system. And I was very grateful to work with the labs Alice Ting, Ben Corvat, and Corey that I had all these training and they also encouraged me to start taking up these additional questions and problems. And I think my past training in chemistry really helped me to think sort of broadly about building new tools. And this compact nervous system of C. elegans really pushed me to think about at a single cell level, what are the cell type diversity in the mammalian brains? And obviously having more precise tools as well as having more sort of higher resolution readout are important for understanding neuropsychiatric disorders and illnesses as we were discussing earlier.
Yeah, what a great trajectory, how you turned your expertise in chemistry into something very useful for people that can change many lives down the line. Through these advances you're making. Thank you for applying your expertise to this work.
I'm very fortunate to work on this very exciting and hopefully meaningful and direction that I can help people.
Yeah, that's wonderful. In particular, what will the funding you receive from the Rising Star Award enable you to do? And what are your long-term and short-term even goals for the research with the Rising Star Award funds?
Yeah, we're very fortunate to receive this award that we will continue working on certain directions that we are currently already undertaking in the lab. And one direction that I'm very excited about is to continue work with the human geneticist, in this case, Stephen Sanders, who is now in Oxford University, our long-term collaborators. And he is really one of the driving force among all these risk gene sort of identification of autism spectrum disorders and neurodevelopmental delay, as well as additional psychiatric illnesses. So working with him, we are interested in expanding the rapporteur of the genetic screen, that previously in the paper we published, we started with 35 risk genes. We're hoping to expand this to cover all these high fidelity risk gene variants that has been identified. And I think the paper literally just came out this month. So you will see that this is a very exciting time that we start gaining deeper and deeper understanding about these risk genes. And at the same time, we want to perform these functional studies using perturbsy to understand how they act, what brain region, what cellular specificity that they act on, and are there any possible convergence? And in the future, of course, we are very excited in the long term to extend this approach to additional gene variants and also to additional genes that's implicated in additional disorders such as bipolar disorder. And this is one of the other directions that the geneticists are making so much progress just in the past 5 years. And we're looking forward to collaborate with them and extend our platform to be not dedicated to just one disorder, but being a generalizable approach to understand how different disorder risk genes may even overlap at a cellular mechanism, which I think would be a very interesting way to think about it. And in terms of these long-term understanding for the entire field of neuropsychiatric illnesses, I think will be really interesting if we are at a stage that we can use this genetic information as well as the genetic mechanism from this risk gene that hopefully we identify, and going forward to be able to stratify certain disorders using these molecular genetic information. And in many other disease and disorder, people talk a lot about the personalized medicine. Yeah. And I think in the neuropsychiatric illnesses, this is also in direction, maybe not so much at a one person at a time kind of personalized medicine, but rather in the group way. If we know that among these 250 risk genes, maybe 30 or 40 of them targeting one cell type, and these are obviously a very interesting way to think about classifying patients and conditions under this broad umbrella of disorder, but being able to understand them in groups, and potentially this will help us to think about additional drug targets and really working with others to perform drug screens and appropriate models. That will be a direction that hopefully in 10 or 20 years we will be able to see the fruition in the field.
That is so exciting, thinking about how you can help people who are struggling with mental illnesses and serious challenges to get the right treatment from the get-go, from the very beginning, and not have to go through months of trial and error and trying different treatments and not knowing kind of blindly which ones might be able to help them or not. I live with schizophrenia, and when I was in my first episode, it took me about 6 months working with my psychiatrist to finally find a medication that would help me recover well enough to go back to school and go on with my life. If we could circumvent that long, fraught process with personalized medicine like you're helping to develop, that will really change a lot of lives. Yeah, well done. Yeah, definitely. So finally, looking ahead to where your research is going, what gives you hope?
Yeah, what gives my hope? I think I am hopeful, very much so. First of all, I would say, the general field has been getting a lot of attention that it totally deserves. I feel like, 10 years ago, we are not so actively talking about mental illnesses or even the genetic basis of these devastating disorders. So I think collectively, the attention that we're getting from not only the federal government funding agency, but also from foundations like One Mind, and also all these patient cohort, there are more and more sort of self-organized citizen science kind of communities started to be sort of visible and start advocating for these very complex problems, but also attracting scientists from all over the field to work on this. I think the energy and the vibrant sort of community really give me hope that we're getting the talents, the attention, the funding, the resources to tackle these questions. And I would say scientifically, I'm also very hopeful because just look at how much we already learned in the past 20 years. 20 years ago, we would not think about a genetic variance can contribute to the manifestation of mental illnesses. We even just 10 years ago, do not have this tool to look at single cell resolution, one cell at a time. What are the cellular compositions? And 20 years ago, we definitely don't have these tools to look at whole brain imaging. These all sounds like magic and poetry 20 years ago, but these are now the tools that we use routinely in the lab. So we are definitely moving forward, and the development of technology and the advancement of sequencing technology, they're not even slowing down. So I would say, given what we have already learned in the past 20 years, I'm very hopeful that in the next 20 years, we're going to learn more. And hopefully we'll be able to translate some of these understanding to really be able to help give it back to the community.
Yeah, that's phenomenal. Science has always given me hope for a better future for people like me who have struggled with mental illnesses. And your work is just an amazing advance in this field. Every year I am blown away by the work of our Rising Star awardees, and this is an amplification of that, just learning about what you're doing. So again, thank you for all that you are doing. It will mean so much for people who are struggling with mental illnesses. Thank you, Xin, for giving hope to so many through your innovative work. Best of luck with your innovative research. Thanks for being with us today on Brainwaves.
Thank you, Brandon.
And viewers, thank you too. Thanks for watching. Thanks for participating. If you have any questions, feel free to post them in the chat still. But you can always check out all of our past Brainwaves episodes at onemind.org/brainwaves, where you can also sign up for our newsletter to be apprised of new episodes coming out. Remember, what is your brain good for? Making waves, of course. Thanks for joining us on Brainwaves today. Have a great day.
Most of the time, I just want to lay in bed. I don't want to talk to people.
Mental illness is the world's leading cause of disability. And right now, our mental health is being put to the test. Who is leading the discoveries to provide quality mental health care from the lab to the front lines? We are One Mind, accelerating brain health for all. Help us fund new treatments at onemind.org.
I'm living my best.